Okay, welcome everyone to the last campus research colloquium of this quarter. We will have a few meetings during spring, but this is the last one before the week of finals and spring break. And what better way than to end the quarter with today's talk. It's my great pleasure to introduce Theanne Griffith from the Department of Physiology and Membrane Biology. And Dr. Griffith will tell us a little bit about herself and about her exciting research. Thank you so much.
All right. Well, hello, everybody. Thank you for joining in today. Today, the title of my talk is Illuminating New Roles for Nav1.1 in Peripheral Sensory Neuron Function. And so, oops, let me get my... All right, so just a little brief outline of the talk today. I'm first going to tell you all just a little bit about myself and the lab and my career trajectory. Then we'll do a little bit of a somatosensory dorsal root ganglion neuron recap for those in the audience who don't regularly think about sensory neurons or our peripheral nervous system. Then we're going to talk about some newly discovered roles for Nav1.1 in both thermosensation as well as proprioreception and motor coordination, and then end with some future directions and why I think you should care about the peripheral nervous system. So just a little bit about me. Won't start all the way from the beginning, but I will start as an undergrad. I earned my bachelor's in neuroscience and Spanish as an undergrad at Smith College. And Smith College is a small women's liberal arts school in Western Massachusetts. And although it is a small liberal arts school, it actually has a very vivacious and lively research program, including one of the largest animal facilities that an undergrad institution has, especially a liberal arts one. So I was really lucky to be able to conduct research all 4 years as an undergrad. And this is where I really fell in love with ion channels and electrophysiology. This is where I was introduced to the Xenopus laevis oocyte expression system. And in particular, I investigated human GABA-A receptor modulation by novel anesthetics using 2-voltage electrode clamp recordings and different in vivo behavioral assays. And so this is some of the data that I collected here. And this was, I believe this was my junior year in college, and this was our group. That was my PI, Adam. And I just wanted to show this picture because I was really fortunate. Although Smith is a predominantly white institution, my lab, the lab that I was in for 2 of the 4 years, was predominantly Black. And that was pretty cool. And I think a little bit kind of inspiring or motivating, or I'm not exactly sure of the correct word, but it was really nice to begin my research career surrounded by other Black women who were also excited about neuroscience. Okay, so after Smith College, I briefly worked in actually South America for a couple years in Chile before heading to Northwestern, where I earned my PhD in neuroscience. And there I switched from studying GABAergic inhibitory ion channels to glutamatergic excitatory ion channels. So my project looked at understanding how ionotropic glutamate receptors, which are pictured down here, are modulated by auxiliary subunits. And so I used this fun technique called fast application, where you can actually pick up cells that are expressing these glutamate receptors and put them in front of this piezo-driven flow pipe to very quickly apply agonists, and we can evoke currents that look like this. And I was able to identify this region within the kainate receptor, these little red linkers here that are really critical for this modulation of these kainate receptors by these auxiliary proteins, NETO1 and NETO2. So I kind of continued down this trajectory of ion channels and really gained, obviously, a much deeper appreciation and understanding of them as a graduate student than I did as an undergrad. But I became more curious, not just about their intrinsic biophysical mechanisms, but how those biophysical mechanisms were contributing to overall neuronal function. So I really wanted to understand how these kind of nifty little molecular machines were operating in a physiological context to confer distinct excitability features to different neuronal populations. And this is what I studied as a postdoc with Ellen Lumpkin at Columbia University. And so I switched my ion channel team again. I went from inhibitory ligand-gated channels to excitatory ligand-gated channels, and finally landed on voltage-gated sodium channels. And I won't tell you so much about that project because I'm going to talk more about that in the research portion of this talk. So now, as an assistant professor here at UC Davis, my lab opened its doors in October of 2020, so smack dab in the middle of the pandemic, but it's been really fun getting things up and running and just seeing the progression of the lab over the last year and a half. It's really kind of come together, and it's really been gratifying to watch. And we use a wide range of techniques that span from ion channel biophysics to animal behavior to investigate how our body encodes and transmits somatic sensations. And there are two kind of overarching questions that we're interested in asking in the Griffith Lab. And the first one of those questions is how are ion channels differentially engaged to transmit discrete somatosensory modalities? And so currently we're investigating roles for understudied ion channels in the peripheral nervous system. And specifically, we have a project looking at voltage-gated sodium channels in proprioreception, which I'm going to tell you about in the second part of the research talk, voltage-gated sodium channels in thermosensation, as well as pain. And then also in a collaboration with my partner, Jorge Contreras, we're very interested in understanding how large pore channels like connexins and pannexins contribute to the development and maintenance of pain and neuroinflammation. And so we also have a kind of newer line of investigation that we're very kind of recently getting off the ground, but we're also interested in this peripheral central nervous system crosstalk and how aberrant peripheral nervous system function could impact brain function. And specifically, we're interested in the contribution of human sodium channel mutations in peripheral sensory neurons to the clinical manifestations or comorbidities that are typically associated with brain disorders, such as epilepsy or autism. And I'm not going to talk about this today because there's not too much to talk about at this point. As I said, we're recently launching this, but I will elaborate at the very last slide a little bit more about this idea. Okay. So lastly, in addition to my work as a scientist, I also write children's books for kids. I feel like you've probably already heard about this, so I won't spend too much time talking about it, besides to say that I'm the author of the science adventure series, The Magnificent Makers. There are currently 5 books out in that series. And I also write the nonfiction Ada Twist Scientist Y-Files series, which accompanies the Netflix show of the same name. And a quick little review or reveal, I should say, before I do this publicly, but there's also a 6th book out in the Magnificent Makers series that I'm really excited about called Storm Chasers. That's going to be coming out next November. Okay, so the first project that I'm going to tell you all about today was a project that I worked on as a postdoc back at Columbia, where I was investigating excitability in peripheral sensory neurons and trying to understand mechanisms by which excitability was regulated in a subtype, in a neuronal subtype specific manner. So just to kind of get everybody up to speed, because I know we're all coming from different science backgrounds, I wanted to give a little overview on what is somatosensation and what are somatic sensations. So somatic sensations are the sensations that we largely perceive through our skin. So pain is a good example of that, as is touch. Itch is also a somatic sensation. When a mosquito bites you, that activates itch-sensitive neurons that transmit that signal to the brain. Also temperature as well. Our ability to detect temperature is a really critical somatic sensation, and that's the one I'm going to talk a little bit more about today. And then also proprioreception. So proprioreception is a little bit of a, doesn't really have a place in somatic sensation per se, although it's generally thought of as a somatic sensation. But this is our ability to detect where our limbs are in space. And so we're able to do purposeful movements and coordinated movements thanks to the work of peripheral sensory neurons that transmit proprioceptive signals. And again, I'm going to be talking a little bit more about that in the second part of the talk. So these somatic sensations are transmitted, and perhaps they're transmitted by sensory neurons, and they can be modulated by other cell types. But in the end, it is these dorsal root ganglion neurons that are innervating our neck down that are tasked with transmitting somatic sensations. And so I'm going to be referring to them as DRG neurons throughout the talk, but their full name is dorsal root ganglion neurons. There are also trigeminal neurons that innervate our face and mouth. And we're not actively investigating those at this moment in the lab. So everything I'm going to be showing you today are DRG sensory neurons. So dorsal root ganglion neurons can be very broadly classified based on their anatomy. So they can be grouped in large, medium, or small diameter populations, and form tends to fit function a bit for DRG neurons. So large diameter neurons tend to be mechanosensory in nature and either transmit proprioreceptive or tactile touch information. Medium diameter neurons can be either, pain sensing. Some of them are pain sensing neurons. Some of them are also mechanosensing neurons. And then your small diameter neurons are largely your pain sensing neurons, your temperature sensing neurons, and some itch sensing neurons. And there is one unique class of small diameter mechanosensory neurons that are believed to transmit social touch as well. So that is in reference to their cell bodies, but these DRG are kind of grouped in these little ganglia that line our spinal cord, and an individual cell has an axon that leaves and then bifurcates and sends a projection to the spinal cord, as well as peripheral targets such as the skin, right? So that small, medium diameter classification is largely based on their soma, but as well as their axons. And so large diameter neurons are going to have myelinated axons that have faster conduction velocities. Medium diameter neurons are going to be kind of lightly myelinated, so they're kind of in between. And then small diameter neurons are going to be unmyelinated. And then finally, they can also be distinguished based on their target end organ structures. So an example here, this purple neuron here is a large diameter mechanosensory neuron that's forming what we call a lanceolate ending. And that's just basically a fence-like structure that surrounds hairs and can detect hair movement accordingly. You also have some neurons that make synapse-like contact with skin cells. So this is what my postdoc mentor studied, the Merkel cell neurite complex. They basically make chemical synapses with these Merkel cells, which are skin cells, to transmit discriminative touch information. And then some neurons, which are called free nerve endings, just kind of end in the skin and don't have a very specific structure. And those tend to be your small diameter neurons. So as you can see, sensory neurons are extremely heterogeneous anatomically as well as functionally. So this is a figure from a paper that I really like. What you're looking at are current clamp recordings, and what is shown in these individual lines are the generation of action potentials. And for those of you in the audience without a neuro background, action potentials are just the currency of the nervous system and how the nervous system communicates. And so in this paper, what they did is they performed electrophysiological recordings from isolated DRG neurons that were genetically identifiable. So using mouse genetics, they were able to fluorescently label subsets of DRG neurons based on their molecular identity. And so, for example, this is a non-peptidergic nociceptor, which is just a way to say a subtype of a pain-sensing neuron. And you can see in response to increased stimulation, it's not terribly excitable, which contrasts significantly with proprioreceptors, those neurons tasked with encoding limb position and movement, which in response to increasing current injections just fire like gangbusters, so very highly excitable. And then these LTMRs are just different mechanosensory neuron populations that are tasked with encoding touch. And again, you can see very distinguishing electrical signatures for all of these different sensory neuron subpopulations. And so not only are sensory neurons very heterogeneous at the anatomical level, they're also very heterogeneous at the functional level, which again makes sense because they're tasked with encoding a pretty diverse array of stimuli. And so in order to transmit these signals, DRG neurons express a wide variety of ion channels, which again can be grouped very kind of crudely or grossly into two groups. Transduction channels are going to be tasked with taking that external stimuli and turning it into an initial depolarization. So a good example of those are going to be your trip channels that might be like heat sensitive or cold sensitive, and upon a change in temperature are going to open and give an initial depolarizing stimulus that will then propagate. The piezo channels you might be aware of, both of these channel families recently won David Julius and Ardem Patapoutian the Nobel Prize. So obviously very critical class of channels. But in our lab, what we're more focused on are the transmission machinery that is tasked with sending these signals from the periphery to the central nervous system for further processing. And so these transmission channels are going to be your voltage-gated potassium channels, voltage-gated calcium channels, and voltage-gated sodium channels, which are the channel subtype that I'm going to hone in on here pretty soon. So in Ellen's lab, what I was really interested in understanding was what are some of the cellular mechanisms that mediate excitability in distinct populations of DRG neurons? And in order to ask this question, I really wanted to have a group of neurons that was developmentally related, but still maintained some level of functional diversity. So as I mentioned, DRG neurons are extremely heterogeneous. If you just culture them and plate them on glass and start randomly patching, you could very easily be comparing kind of apples and oranges and have a signal to noise problem. So in order to circumvent this, I turned to a VGLUT3 TD tomato transporter, transgenic reporter line. And so VGLUT3 just stands for vesicular glutamate 3. That's not really particularly important to the story, but that was the marker of this subset of neurons. And as you can see here, looking at the red cells, we're labeling about 20 to 25% of all DRG, so a developmentally restricted population. And as you can see here in this red channel, you can see some small diameter neurons, some more medium diameter neurons, as well as large diameter neurons, suggesting that there is some functional diversity as well. And indeed, these VGLUT3 lineage neurons had been implicated not only in different forms of touch, both gentle touch and discriminative touch, but as also pain, so nociception, as well as cold sensing. So they were a restricted population that maintained some level of functional heterogeneity. And so it really served as an ideal model to try and tease apart mechanisms of excitability. And for the sake of time, I'm not really going to show you exactly how I got there, but what we discovered was that tryptamate positive cold receptors, which we identified based on their sensitivity to the cooling compound menthol. So menthol, as you all know, is a compound derived from mint. It's in a lot of things, mojitos, toothpaste, menthol is kind of everywhere, but it does give you this kind of cool sensation. And that is because it activates this cold sensitive ion channel, tryptamate. And so what we found was that menthol application selectively activated small diameter VGLUT3 lineage neurons. And so what we decided to do was focus in on small diameter VGLUT3 lineage neurons that were menthol sensitive or insensitive and compare those two populations to see if we could find distinguishing excitability mechanisms. And so I turned to in vitro patch clamp, my bread and butter. And so here you can see we have a fluorescently labeled small diameter DRG neuron. And when we applied menthol to these cells, we found that about half of them fired action potentials in a dose-dependent manner. So you can see that quantified here. And so this was really exciting because if we could target small diameter V-GLUT3 lineage neurons, they were clearly pretty excitable. And I'm going to talk about that a little more in the next slide. But then about half of them were these TRPM8 positive menthol sensitive neurons. And the other half were kind of probably a smorgasbord of nociceptors and maybe some mechanoreceptors as well. Oops. Oh, I'm missing something here. Sorry, I'm missing a slide here. I had something originally that said how we did the recordings moving forward. So all the data that I'm going to show you, and maybe this is on the next slide, so sorry if I repeat this, but all of the data that I'm going to show you was obtained from adult mice, so 6 to 16 weeks of both sexes. And we targeted these small diameter V-GLUT3 lineage neurons and then determined whether or not they were menthol sensitive or not and classified them accordingly. And none of our cultures were performed with any growth factors or anything. For those of you who are familiar with DRG neurons, sometimes people include growth factors, but that can change ion channel expression a bit. So we did acutely dissociated, and cells were recorded within 24 hours of plating. Okay, so one thing that we noticed about these menthol sensitive cells is they were in fact really excitable, and at room temperature they already exhibited very noticeable firing patterns, either the sustained firing or more of a phasic firing pattern. But for anyone who's recorded from DRG neurons in culture, you know that typically in the absence of any kind of stimulation, these neurons are very quiet. You're not going to just see this kind of firing that I observed. So that was great because it again showed that these neurons were really excitable. We observed no firing, no spontaneous firing in the menthol insensitive population. So it really gave us a nice group to compare with. We did realize, though, that because we were doing these recordings at room temperature, it was very likely that this spontaneous firing was actually temperature evoked firing. And so to test this, we applied a blocker, this PBMC compound, which blocks TRPM8 channels, those cold sensitive ion channels. And indeed, when we blocked TRPM8, we were able to inhibit that action potential firing. And so what that suggests is that firing, that room temperature spontaneous firing, is dependent upon TRPM8, and the neurons that I'm recording from are probably overwhelmingly the menthol sensitive neurons, TRPM8 expressing cold receptors. Yes, that's what I thought. Sorry. Okay, so there's the information about the recordings that I explained in the previous slide. Sorry, I got those backwards. Okay, so the first thing that we did were some basic current clamp analysis. So we evoked action potentials in menthol sensitive and menthol insensitive neurons and quantified various parameters. It was very evident that they fired more action potentials. We also noticed changes in their action potential waveforms. The menthol sensitive population action potentials were much skinnier, which makes sense because it allows them to fire more. They also lack this classic hump in the repolarization phase. So this hump is very typical of nociceptors, and it's believed to be due to the complement of ion channels that they possess. So this already is giving us some kind of idea that there are differential mechanisms involved here. But there were two parameters that I found really striking. One was their action potential threshold. So the menthol sensitive population action potential threshold, the membrane potential at which you're able to generate an action potential, was significantly more hyperpolarized than the menthol insensitive population. And then furthermore, their resting membrane potential was actually more depolarized than the menthol insensitive population. So they were kind of sitting at a membrane potential that really closely bordered their action potential threshold. So it took very little stimulus. That's kind of the idea. It would take very little stimulus for these neurons to generate electrical activity. All again consistent with an excitable neuron population. But this really had me very curious because for anyone who is an electrophysiologist or who studies neurons in general, you might note that this is a pretty depolarized resting membrane potential. And I will say that DRG neurons in culture just tend to have very depolarized resting membrane potentials as compared to central neurons, which are very happy at like your typical -70. These guys sit at around -50, but these were particularly depolarized. And the interesting thing is that they were still able to maintain action potential firing. So I was really curious about the voltage gated sodium channels that were allowing them to maintain this kind of activity. Voltage gated sodium channels will definitely set your action potential threshold, and sodium channel inactivation caused by depolarization can actually reduce excitability. So I was curious as to whether or not there was something going on with the voltage gated sodium channels in these neurons. So just a quick recap, voltage-gated sodium channels are critical mediators of excitability. They're responsible for the initial depolarization phase of the action potential. And in mammals, we have 9 different isoforms, Nav1.1 through Nav1.9. And in sensory neurons, in adult sensory neurons, you have 5 of those isoforms, Nav1.1, 1.6, 1.7, and 1.8. And there's a little bit of dogma in the field that Nav1.1 and 1.6 primarily contribute to firing in those large diameter mechanosensory neurons, whereas some combination of 1.7, 1.8, and 1.9 mediate firing in the small diameter population, like these menthol sensitive neurons. And indeed, one of the first studies to look at sodium channels in cold sensing neurons attributed excitability to the Nav1.8 isoform. So what they did here is they recorded action potentials from neurons from either wild type or Nav1.8 knockout animals at either 30 or 10 degrees, and found that the knockouts were unable to elicit action potentials at 10 degrees. And so this led to the idea that Nav1.8 was really critical for action potential firing at cold temperatures. I won't get into all the background on it, but I will say that that has pretty much been debunked a little bit in the years since this paper's initial publication. But nonetheless, we decided to take a pharmacological approach to identify the sodium channels that were responsible for action potential firing in our menthol sensitive population. And to do this, we first turned to a very tried and tested sodium channel inhibitor, TTX, tetrodotoxin. This is going to inhibit Nav1.1, 1.6, and 1.7. And surprisingly, contrary to dogma, we found that inhibiting these 3 channels was sufficient to inhibit completely action potential firing in menthol sensitive neurons. And so that means that some complement of those 3 channels is responsible for mediating action potential firing in those. And so I'll just kind of cut to the chase and say that we were able to rule out a role for Nav1.7. We lack the pharmacological tools to really look at Nav1.6. But when we applied an Nav1.1 inhibitor, we found that the vast majority of action potential firing was abolished, which you can see visually here and quantified here. So this was really exciting because no one had thought that Nav1.1 had anything to do with thermosensation or transmitting electrical signals in cold sensitive neurons. So that was a pretty exciting finding. So we kind of identified the character, the culprit, Nav1.1, but as a person trained in ion channel biophysics, I really wanted to know the mechanisms through which Nav1.1 was contributing to excitability. So I'm just going to quickly run you guys through the sodium channel gating cycle so you have a little bit of background for the next slides I'm going to show you. So sodium channels at rest will be in the closed conformation, and upon depolarization will open. And depending on how strong and how long that depolarization is, they will either enter into fast or slow inactivation. So fast inactivation follows more short stimuli, and it occurs very quickly on the order of milliseconds, but then recovery from fast inactivation also occurs on the order of milliseconds. Conversely, slow inactivation takes a little bit longer to enter into, maybe 100 or 100 plus milliseconds. But recovery from slow inactivation is also really slow, even on the order of seconds at times. And so slow inactivation is thought to work as an excitability brake and really dampen down action potential firing in certain neurons. And that was showed here by Bruce Bean and colleagues almost 20 years ago now, where they found that in certain pain sensing neurons, as the action potential firing reduced or adapted, you saw an increase in the amount of slow inactivation. So we asked the basic question, we have a neuronal population that's super excitable. Perhaps they're refractory from entering, the sodium channels in those neurons are refractory from entering into the slow inactivated state. So that was the question that I set out to address. And indeed, they were. So that was really exciting. So when we recorded from menthol sensitive neurons and we ran our entry into slow inactivation protocol, what we found was that even after over a second and a half of a depolarizing stimulus, about 80% of the current was still left over. So that means that only 20% of the sodium channels were kind of taken out of the picture, and the other 80% were free to continue eliciting action potentials. This contrasted with the menthol insensitive population, where you can see the sodium channels in those cells really readily went into the slow inactivated state. So this was cool because it provided perhaps a biophysical mechanism that explained the heightened excitability of these menthol sensitive neurons. But then we obviously wanted to know, what role is Nav1.1 playing in this? So we were able to use this ICA compound to isolate out the Nav1.1 sensitive component of the current, and found that it was about 38% of the total. And importantly, when we blocked this component of the current and then recorded slow inactivation kinetics for the remaining residual sodium current, that current readily went into the slow inactivated state. So that's what you can see here in these dark filled circles, and these clear circles are the same data from over here. So just the whole cell sodium current. So this was really exciting because it meant that perhaps Nav1.1 is responsible for the fact that the whole cell sodium current is largely not entering into the slow inactivated state. And that again might be a mechanism by which these neurons are able to maintain firing. And we also did a correlation where we looked at the magnitude of the Nav1.1 mediated current. And when you had a high percentage of the sodium current carried by Nav1.1, what we found is that when you block that, the tau for entry into slow inactivation was a lot faster than when the overall contribution of Nav1.1 to the whole cell sodium current was lower. And that was a significant correlation, indicating again that Nav1.1 might be driving the entry into slow inactivation kinetics and thereby promoting neuronal excitability. And so this was the model that we worked with, whereby you have activation of TRPM8, which leads to activation of these TTX sensitive sodium channels. We couldn't really rule out a role for Nav1.6, but we could say that Nav1.1 appears largely to cycle through these fast inactivated states pretty rapidly without being sequestered into the long-lived slow inactivated states. And that's what allows for repetitive action potential firing. So I'm running a little bit long, so I'm going to kind of skip over some of these future directions and open questions and just go straight into the second part of the talk, which is looking at a role for Nav1.1 in other sensory modalities. So Nav1.1 in the peripheral nervous system is fairly understudied, and I think that's largely due to its prominent role in brain function. So Nav1.1 has widespread expression throughout the central nervous system. It's found in the cerebellum, hippocampus, and cortex. And importantly, the gene that encodes Nav1.1 is referred to as a super culprit gene. And that's due to the hundreds, and I think over 1,000 now, disease-causing mutations that are associated with its locus. So diseases like epilepsy, autism, and even migraine have been attributed to Nav1.1 dysfunction, with I think epilepsy probably being the most notable and the most represented among all of the diseases. And it's very likely that this very prominent role that Nav1.1 plays in brain function has taken some attention away from the also important roles that it could be playing in the peripheral nervous system. And so there's really not a lot of data out there, and probably some of the best data is from this paper that was published by David Julius's group. And in this paper, what they found was that in the periphery, Nav1.1 seems to play a role in non-inflammatory pain. And so what they did here was they injected the hind paw of a mouse with this activator. This HM1A is an activator of Nav1.1. And they injected that into either wild type animals or animals that were missing Nav1.1 in some of their DRG neurons. And what they found is that in wild type animals, activating Nav1.1 elicited pain-like behaviors, except in the knockout animal. And then this pain behavior, these were spontaneous pain behaviors. When they specifically looked at heat pain, they didn't see any differences between the wild type and the knockout upon activation of Nav1.1, suggesting that Nav1.1 is not involved in heat pain. But when they looked at mechanical pain, they did in fact see that when you injected animals with this Nav1.1 activator, their threshold, their ability to withstand touch was much lower than knockout animals, again, suggesting a role for Nav1.1 in mechanical pain. But we wanted to know, what else could it be doing? That's just one study. There's a handful more. They all kind of tend to focus on mechanical pain. But we wanted to understand what else Nav1.1 could be doing in the peripheral nervous system. So Cheyenne Lewis, who is a junior specialist in the lab, performed a series of RNA scope experiments. And what we did here is we labeled different populations of sensory neurons. So in this top panel, we're looking at peptidergic pain sensing neurons. And in this bottom panel, we're looking at non-peptidergic pain sensing neurons. So just two different kinds of pain sensing neurons. And what we found was not that much expression. I'll show you the quantification in a second. Conversely, when we looked at this bottom panel here, we have our myelinated neurons, so all of those large diameter mechanosensory neurons. The vast majority of those, I think about 93%, were positive for Nav1.1 mRNA. And then when we took a transgenic mouse that labeled proprioreceptors with TD tomato, we actually found that 100% of proprioreceptors expressed Nav1.1. So this was, a very curious observation. If 100% of the neurons are expressing mRNA message, might there be some function for the channel in these neurons? So just a kind of brief overview of proprioreception. As I mentioned earlier, it's kind of a little bit of a funky somatic sensation. It's often referred to as a sixth sense, because while I would argue that a lot of our somatic sensations we don't think too much about, proprioreception we really think little about. It's just kind of our autopilot. And so again, this information is carried by these large diameter DRG neurons that innervate skeletal muscle as well as joints and tendons. And in skeletal muscle, they form these really beautiful end organs called muscle spindles that detect muscle stretch. They also form different kinds of end organs that also detect force. So the transduction channel that's really critical for proprioceptive signaling is also piezo2, which is, again, that touch-sensitive channel that was identified by Ardem Pataputian's group. But what is unclear is whether or not Nav1.1 or any sodium channel, the identity of any of the sodium channels that transmit proprioceptive information, is pretty unknown. But there is evidence that Nav1.1, in addition to having message be present in dorsal root ganglion soma, of proprioreceptors. That's also found, protein was also found in these muscle spindle afferent endings, suggesting that there might indeed be a functional role for this channel in proprioceptive signaling. So last summer at Woods Hole, we performed some really fun experiments with I was a teacher in the neurobiology course there, and we performed some really fun experiments with 2 students. So they did these current clamp experiments, very similar to the data that I showed you previously, where they evoked action potentials and then they inhibited Nav1.1 with this ICA compound, and indeed did find a significant reduction in action potential firing, as well as action potential amplitude. The threshold was changed, as did the width of the action potential. And all of those changes are very consistent with loss of Nav1.1 function. And so more recently, back in the lab, a graduate student, Cyrus Espino, has performed some voltage clamp recordings. And here we see that on average, Nav1.1 seems to contribute to about 50% of the whole cell sodium current, which again makes sense with its role in controlling neuronal excitability in proprioreceptors. So this was all fine and dandy, but in the end, it's in vitro and using pharmacology. So we set out to make a mouse that would lack Nav1.1 in proprioreceptors. Unfortunately, due to the fact that the marker for proprioreceptors is the same as the marker for many brain neurons, in which Nav1.1 plays a critical role, we were unable to restrict our deletion of Nav1.1 to just proprioreceptors, but we were able to use a different mouse model from the one I showed you in the Julius paper, in which Nav1.1 is deleted in all sensory neurons. And we were pleasantly surprised to find that these mice had a very noticeable motor phenotype. So I'm going to play a couple videos for you here. They may not be so obvious if you're not used to looking at mice, but what you'll notice is that when these mice are suspended in the air, they shake their legs in a very ataxic manner, which is not normal, and they do a lot of very abnormal limb positioning, which you can kind of glance from these 2 pictures. So normally when you hang a mouse by its tail, it's kind of trying to get a sense of balance and tends to splay its hind paws, whereas our conditional knockout animal exhibits a clasping phenotype, which is very characteristic of mice that have some kind of motor dysfunction or deficit. So here's an example. You can see this mouse here on the left is clearly exhibiting this kind of ataxic-like behavior. Oops, sorry about that. I played the video again. And so there we go. So here's another mouse, and you can see it's kind of shaking. It's unable to kind of coordinate, but at the same time, they do have a certain amount of strength. As you can see here, they're able to pick up their food pellet just fine. You're going to notice at the very end of this video a very strange limb positioning about right here, where the mouse just completely turns its hind paw around to grab its tail, which again is not normal at all. And then here, as I'm scruffing a mouse, again, you'll see that its paws are completely turned around. So you see its front paws are kind of palms up, where its back paws are completely twisted. So a clear lack of sense of limb position. We quantified this in the open field test. And interestingly, in the open field test, we found out that both HETs and the conditional knockout animals both moved slower as well as moved less in the open field assay. When we looked at the rotarod assay, which basically asked how long mice can maintain their position on a spinning beam, we found that there, the HETs performed very similarly to the floxed animals. However, the conditional knockouts were very, very poor at performing this task. They clearly lacked motor coordination. And so while the HETs also have some motor phenotype, it's not quite as severe as the conditional knockout. So we then wanted to make sure that our conditional knockout was indeed just targeting DRG. I had read somewhere in the literature that some motor neurons might express PERT, which is the promoter that's driving Cre expression in our model. But what we see here when we crossed our PERT-CRE line to a TD tomato reporter, this AI14 line, we can see our DRG lit up beautifully with very few cells found to express TD tomato in the spinal cord. So we're quite confident that our knockout model is indeed restricting deletion of Nav1.1 to sensory neurons. We are collaborating with Dr. Catherine Wilkinson at San Jose State University. She does muscle afferent ex vivo recordings where you can take out a muscle and record from the proprioreceptive axons and measure action potential firing. So the data haven't been fully analyzed, but here are some example traces. And so as you can see in this floxed example, this is a very characteristic proprioreceptor response where you have kind of an initial kind of burst of firing that is then maintained for the duration of muscle stretch. So that's what this is representing here, the degree of muscle stretch. The het animals, and this is a little bit anecdotal since I don't have the summary data, but in my conversations with Katie, it seems that the het animals kind of look like either a knockout or a floxed animal, but seem to have this kind of intermediate phenotype, which is consistent with our behavioral analysis. And then our conditional knockouts, importantly, seem to have a deficit in their ability to maintain firing for the duration of stretch. So again, I don't have any quantification for this at the moment. She's working on that and we should have that pretty soon, but we think that this pattern is going to hold. Okay. So Cyrus also did some immunostaining of the muscle spindles in our different groups to be sure that we weren't having some aberrant developmental effects due to loss of Nav1.1. And qualitatively speaking, we haven't observed any major deficits in muscle spindle formation. And this isn't wholly surprising because the piezo2 knockout animals, which also have major proprioreceptive deficits, perhaps even more than our knockout, also have intact muscle spindles. So we don't think that any of our behavioral phenotypes are due to muscle spindle abnormalities in muscle spindle development. We think it's definitely an electrical signaling deficit. However, it could also lack of innervation into the spinal cord could also be an issue in our model. So what I'm showing you here are sections of spinal cord, of the ventral spinal cord from a floxed animal and a conditional knockout. And what Cyrus did here was he stained this for VGLUT1, which is going to label your proprioreceptive axon boutons, as well as CHAT, which is going to label motor neurons in the ventral spinal cord. And so proprioreceptive neurons make synapses onto motor neurons. And what we found here was that there was a significant reduction in the number of puncta per cell per CHAT positive motor neuron in the knockout compared to the flox. And I don't have the data up here, but he recently quantified the het data and found no significant difference between the flox and the het data. So this seems to be something that occurs only in the knockout and could represent some kind of activity dependent decrease in innervation, perhaps, of the spinal cord. And so this altered spinal circuitry could indeed be contributing to our overall phenotype. We don't think this lack of innervation is arising or is occurring, because of reduced proprioceptive number. So Cheyenne performed some RNA scope experiments where she labeled sections from either floxed or conditional knockout animals and probed for parvalbumin and RUNX3. And co-expression of those two markers is kind of a true and tested way of identifying proprioreceptors molecularly. And we found, we found no significant difference in the number of proprioreceptors between flox animals and the conditional knockouts. And again, the data is very new, so I don't have it in here, but we also didn't see any difference with the hets. So we don't think that there's any developmental decrease in the number of proprioreceptors. We think that the lack of innervation is coming from an activity dependent, cells aren't firing so well together, so they're not wiring so well together. Okay, and so with that, I'm going to leave you with some take-home points. We found that Nav1.1 contributes to repetitive firing in proprioreceptors. And so we're very, very interested in what role other sodium channels expressed in proprioreceptors play. We currently have, so proprioreceptors do express Nav1.6 and Nav1.7, and we currently have the Nav1.6 floxed animal. So we're going to be crossing that and creating a very similar knockout as to what you see here, and asking to what extent it phenocopies our Nav1.1 knockout. And then Nav1.1 expression in sensory neurons is clearly required for normal limb position and motor coordination. And so we're very curious as to what degree is the abnormal spinal circuitry contributing to our observed motor deficits. We only looked at one population of motor neurons, but there are several other cell types that proprioreceptors synapse onto. So we're very eager to kind of characterize that a little bit more thoroughly. And then also, Nav1.1 is likely to be expressed in tactile sensory neurons, and it's unclear in our model if abnormal touch sensitivity has anything to do with this motor phenotype. And then we're also very curious as to the temporal requirement for Nav1.1 to proprioreception. In other words, can we knock it out in adulthood and produce a similar phenotype? And then finally, loss of Nav1.1 in sensory neurons is sufficient to alter spinal cord circuitry, which again means that loss of Nav1.1 in the periphery is sufficient to induce a change in central nervous system circuits. And so we're really interested, if we already have a deficiency in the spinal cord, is that relayed up to motor centers in the brain? And in other words, does loss of Nav1.1 in the peripheral nervous system affect brain function? And I know I'm running a little bit long, so I think I'm just going to kind of quickly, unfortunately, skip over this last slide and just say thank you. Thank you to the wonderful members of the Griffith Lab, as well as the Contreras Lab. Thanks to the SAC Lab, who shared some mice with us, as well as a shout out to the neurobiology course from the Marine Biological Lab, where some of this work was done, as well as the Wilkinson Lab in San Jose State. And so with that, I will take any questions you might have.
Thank you very much.
Sorry for running long.
No, you're fine. Thank you for a fascinating talk. If there are questions, please just unmute yourselves. And it is tradition that we stay on until 4:30 if people want to have an extended Q&A or just informal conversation. So there will be time to show that last slide that you
Okay, yeah, I can come back to that if people are curious. I wasn't sure.
Let's take questions first.
I have a question. Yeah.
Hey.
All right, let me lower my hand. Very nice talk. Super cool stuff. And it's funny because, I studied the cells that make the cells you study, but I still don't know enough about the cells you study.
We should talk.
I mean, so this is actually one of the questions that I have is how early is how early is this expressed during development? Is it not until they're differentiated channels, or is it earlier?
So are you talking about how early is Nav1.1?
Yes.
I'm actually not 100% sure of that answer. What I can tell you is that proprioreceptor molecular identity does not fully develop until walking behavior develops. So there was a really nice RNA sequencing study that kind of looked at this. And what they found is that proprioreceptors at P5 basically are like not molecularly identifiable. At P12, they begin to emerge, and then by 8 weeks, they're solidly mature. So I don't know what's going on between P12 and P56. There's like kind of a big gap there, but they definitely don't fully molecularly develop until a little bit later. Now, at what point does Nav1.1 become critical for the firing is exactly what we're hoping to tackle in that collaborative NSF that I was talking about or before we all kind of got started. So that's an excellent question. And I think the jury's still out and we hope to be able to address that.
Awesome. Thank you.
Thank you. Stop sharing my screen so I can see.
Other questions, please feel free to unmute yourselves. well, that's my question if no one else is jumping in. You talk about molecular identity of proprioreceptors and about how proprioreception is like the sixth sense. So the question is, how well defined is it in general and at the molecular level, and how easy is it to detect those markers?
So I would say that our understanding of proprioreceptor development is not very strong. I'm actually really excited that we kind of stumbled upon this project. I know I kind of explained it as very hypothesis driven, but I actually made this mouse to look at thermosensation because of my postdoc work, and then found that it had this major motor phenotype. So there's not a ton known, I would say, about proprioreceptor electrical properties, proprioreceptor development. There's obviously some. Most of the work has been done using these ex vivo preps. And the limitation of those is you can't get kind of mechanistic information. You can compare knockouts and you can use pharmacological approaches, but you can't do some of the, like, we can't ask questions about the properties of sodium channels that are conferring these different electrical properties using these ex vivo approaches. We really need patch clamp to do that. So I would say that there's actually a lot of room to study things ask these kind of questions. And so that's why I'm really excited to have kind of stumbled into this field. I think it's just really wide open. There's a lot to be done. And I'm really lucky that Katie is like right around the corner. I just want to give like San Jose State a shout out, because she works with exclusively like undergrads and master's students and does really, really amazing work. I'm trying to recruit so Serena Ortiz did a lot of the muscle afferent recordings, and she's an undergrad student. And so she's going to be doing her master's with Katie, but then we'll be applying to PhD programs. So I'm definitely going to try and get her in the lab, just because there's, like I said, there's just so much to do there. And in my discussions with Katie as well, she was just really excited to have somebody else that's kind of interested in proprioreceptors to collaborate with, because there aren't that many studies out there. And there are very few studies using patch clamp. And in fact, I think there are like 2 that have used patch clamp, voltage clamp recordings to look at currents in these cells. And there's none that have looked at sodium channels. So that's going to be a quick follow-up paper that we're going to work on now. It's just a nice characterization of the whole cell sodium current with and without Nav1.1. So we're going to try and milk this bad boy.
Yeah, absolutely. No, that sounds really good. And it sounds like a wonderful pipeline from San Jose State to here.
And one thing that I didn't mention either is we also have a floxed animal an auxiliary subunit. So these channels often have auxiliary subunits that are really critical for channel function. And one of the sodium channel subunits, beta 4, is expressed in proprioreceptors. And we have that floxed animal that we're currently running in behavioral analyses to see to what degree that protein also contributes to motor behaviors.
And you were talking about the other receptors, so Nav1.1, but then there's all these other ones. So what would be the next immediate goal?
So the next immediate goal is going to be to analyze at the behavioral and cellular level the contribution of Nav1.6. We're also going to analyze at the behavioral and cellular level the contribution of this beta 4 subunit that I was telling you about. And the other one that's there is Nav1.7. Now, I'm a little bit less interested in Nav1.7, mainly because people have looked at that knockout. Like, you can have a global knockout of Nav1.7, and no one has reported any overt motor phenotype. So I guess that doesn't mean there isn't one, but I'm not super confident that that channel is contributing at the behavioral level to proprioreception or motor behaviors. It's very possible that in a knockout, we might see some electrical deficits, but I would guess that those are not sufficient to lead to behavioral deficits. But we plan to basically dissect out those three proteins, NAV1.6, beta-4, and NAV1.7, using similar approaches to what I showed here, but also kind of extending and getting at these questions of like, when do channels turn on and when do channels become important? I'm also very curious as to where. So that one immunohistochemistry figure that I showed, that's just one study that has done this. No one else has repeated it since. So I don't know how true it is. But their NAV1.1, 1.6, and 1.7 are clearly at the peripheral terminals, but they also could be at the central terminals in the spinal cord, and activity of those channels there could be very important for vesicular release, right? They could be depolarizing that presynaptic terminal that leads to calcium channel depolarization and release of, probably glutamate. These are glutamatergic neurons. And so that, I'm very interested into where along the proprioreceptive axons are the channels expressed and what they're doing peripherally versus centrally as well.
Super interesting. Well,